Now, welcome to session 2. Myself Jalaja Sajeev, PGT Bio from Kendriya Vidyalaya, Keshapuram. In the previous session, I have given you the question to answer. Here is the question for you to think about it and answer. Is there any need for emasculation in a pea plant that is considered as male parent? If not, why? There is no need for emasculation in a pea plant that is considered as male parent. It is because emasculation is done to convert a bisexual flower into a female flower only. So, there is no need to remove the anthers from a bisexual flower if you want to convert it into a male parent. Here the parents, that is P generation, male into female. Phenotype, tall, dwarf. Mendel considered in the original cross the character, the phenotype tall as male and dwarf as female. Now, here comes the question. These pea flowers are bisexual. So, how can it be converted into a male flower? The answer is, there is the technique called as emasculation, that is removal of anthers from a bisexual flower before its dehiscence. The purpose is to avoid self pollination. Our aim is hybridization program, cross pollination. So, we have to avoid self pollination, and for this purpose we are using the technique emasculation, that is removal of anthers from a bisexual flower to convert into a female flower. So, here this particular plant, that is the dwarf plant, is subjected to emasculation in order to convert it into a purely female flower. Now, coming to the genotype, as I have already told you, the dominant character here is tall. Tall begins with the first letter T. So, we will consider and write it as T, capital T for dominant character, and we will write it 2 capital T's because a character is controlled by a pair of genes. Now, accordingly here we have to use small t. The recessive character is controlled by or represented by small letter, and again we will write 2 small t's because a character that is controlled by a pair of factors or genes. Now, next comes the gametes. During gamete formation, the pair of genes, alleles, that will segregate and move to 2 different gametes. So, accordingly we will get 2 gametes here represented as capital T, capital T. Here it is small t and small t. So, when you are writing these gametes, care must be taken to write it in circle. Then coming to the selfing, capital T gamete fuses with small t gamete. We will get capital T, small t alleles together. Now, what will be the phenotype of this particular hybrid individual? This is our F1 hybrid. F1 hybrids, I am representing only one union here, the other progeny is also of same genotype and phenotype. So, capital T and small t alleles. Now, what will be its phenotype? Here comes the question. Capital T is dominant over small t, and this capital T will express its character and small t gets suppressed. So, we know that capital T is meant for tall character. So, the phenotype of this progeny, that will be tall. So, tall in phenotype. Now, the next step is selfing of F1 hybrids. To produce the F2 generation. So, that we can represent as capital T small t into capital T small t. One we will consider as male and the other as female here. Now, next again their gamete formation. What are the different types of gametes produced? So, these two alleles, contrasting alleles, they will segregate and move to two different gametes like this. Now, RC punnet shown this combination of F2 progenies, their genotype, their phenotype and the possibilities in a typical column method, that is known as checkerboard or Punnett square. So, we can represent here the different possible phenotype as well as genotype of these F2 progenies by using columns. Here we will write the male gamete on this side, vertical side, and female gamete on this horizontal side on the top portion. So, capital T one gamete, small t the other gamete. Here again the male gamete capital T and female gamete small t. Now, we can write the combination. Capital T gamete fuses with this female gamete capital T, we will get the combination like this, and this gamete capital T with this small gamete, with the small t gamete female gamete, we will get the other combination capital T and small t. Here again capital T small t. In some cases the students are making the mistake here, please listen carefully. Capital T, that means the dominant allele must be written first, yet another basic concept that everybody must listen. The capital T, that means the dominant letter must be written first, followed by the small letter. Here it is small t, small t. Now, coming to the phenotype, capital T will produce tall. Tall phenotype, capital T and small t, it also produces tall, because as I have already told you, capital T gene meant for tallness is dominant over small t gene for dwarfness. Now, this particular progeny, that is small t progeny, it produces dwarf phenotype. So, we got only 3 different types of genotype here. One capital T that is purely homozygous, tall, and this one is capital T small t that is heterozygous. Again small t is purely homozygous. Here I want to emphasize one more concept, basic concept. A recessive individual will be always homozygous. A recessive individual will be always homozygous. Now, count the number of capital T individuals in this column. You can see only one. Capital T small t individuals, that is 2, and small t individuals will be 1 again. So, here we will get a ratio 1:2:1. This is the genotype. So, this ratio we will call it as genotypic ratio. It shows the different types of possible combinations of alleles and the progenies of the F2 generation and its genotype. So, we will call it as genotypic ratio. So, in a monohybrid cross, the genotypic F2 genotypic ratio will be always 1:2:1. This is yet another basic concept. Now, coming to the phenotype, you can see 2 phenotypes here, one is tall and the other is dwarf. How many individuals are tall? 3 are tall. That means 75% are tall here. So, 3 tall and how many are dwarf? Only 1. So, that means 1 dwarf. So, tall and dwarf again it is phenotype, not the genotype. So, phenotype we are saying, so here we got the ratio 3:1. So, this ratio 3:1, that is known as phenotypic ratio. So, 2 concepts coming here, the basic in genetics, F2 genotypic ratio of a monohybrid cross, that is 1:2:1, and F2 phenotypic ratio of a monohybrid cross, that is 3:1. Now, accordingly, here you can see this particular tall individual and this particular tall individual. There is difference in its allelic constitution. This is having similar alleles, so homozygous, and these two are having different alleles, dissimilar alleles, so they are heterozygous. So, Mendel produced the F3 generation by selfing these progenies, F2 progenies. So, we can see what will happen when we are selfing this capital T progeny here. That means 3 tall progenies are there, out of this 1 tall. That means 1/3 tall. It has the homozygous genotype. When we are selfing it, we are writing like this. One we will consider as male and the other as female. Again the gametes capital T, here also capital T, we will get only similar type of progenies. Only tall progenies. This is the concept that it is homozygous. The same character tall, that is produced in the coming generations, in all the generations on selfing. Now 2 by 3 told progenies of the F2 generation with the genotype capital T small t. If we are selfing this particular individual, it is equivalent to the selfing of the F1 tall hybrids. Even though I will show you capital T small t into capital T small t. Like this, the gametes capital T, this will also separate. Now its union again without any, why we are using this checkerboard to represent this or Punnett square to represent these progenies? Here comes the answer to avoid the errors and confusions in writing the genotype. So here the female gametes we will write here capital T small t, male gametes here capital T small t. Again you can see the result will remains the same as that of the F2 generation. So according to Mendel, the 2 by 3 F2 tall progenies will produce 3 tall and 1 dwarf progeny in F3 generation on selfing. Why this ratio came? Because again the answer is they are heterozygous. Now 1 by 4th of the progenies of F2 generation, that is the dwarf progeny. We know that dwarf is recessive, so its genotype is small t, homozygous one. If we are selfing this particular progeny, then also we will get only one phenotype, that is the dwarf phenotype only. Here it is emphasized why we are getting this same phenotype again and again. The answer comes, it is homozygous, controlled by similar alleles. Now we can go to this yet another concept, that is test cross. Test cross, it means we are using this particular cross to test the genotype of an unknown individual by crossing that particular individual with a recessive parent, or we can define this as crossing of an F1 hybrid back to its recessive parent. This cross is named itself as test cross because it is used to test or identify the genotype of an unknown individual. Suppose in front of us there is a tall individual, we can't say that whether it is homozygous tall or a heterozygous tall. We can't say it as capital T or capital T small t. So in that particular case, if we don't know the genotype of an individual, in order to identify its genotype, we are using this particular cross, test cross. So it is written or known as test cross. And how we will identify this case? Here after the crossing, if you are getting the progenies in an 1:1 ratio regarding the phenotype, then the genotype of the unknown individual will be heterozygous. Here comes the clue. Here this is yet another important basic concept that you have to listen. Suppose in a particular problem it is given the progenies are appearing in 1:1 ratio, identify the genotype of the particular character. So there if it comes in the 1:1 ratio regarding the phenotype, then no doubt about it, it will be a heterozygous individual. I will show you the cross again. Listen carefully. Test cross. For example, in this original cross, tall into dwarf. We know that capital T, here it is small t. The gametes like this, and F1 hybrid is capital T, small t. This F1 hybrid, if we are crossing back to its recessive parent, we are crossing this with its recessive parent. Capital T small t into small t. This is also tall and this one dwarf. We are crossing an F1 hybrid back to its recessive parent. Or suppose we do not know the genotype of this particular individual and we are crossing with its recessive parent. Then gametes like this we will get. We can again show it in a Punnett square. For example, this one is considered as the female parent and here it is considered as the male parent. In the original cross also this is female and this one is male. Here we are writing the female gamete, male gamete here, capital T and small t. Capital T, small t, capital T, small t, small t, small t and small t, small t. Here means these two progenies, they are tall and these two progenies, they are dwarf. 2:2, that means 1:1. Phenotype 1:1. Now comes to the genotype. We are getting only two types of genotype here. One is capital T, small t. The other is small t, small t. 1 and this is also, here it is 2 and 2, that equals 1:1. Can you came to a conclusion here after seeing this ratio? Yes? The ratio, that is phenotypic ratio as well as genotypic ratio, both will remains the same, that is 1:1. So here comes yet another application question. Name a cross in which the phenotypic and genotypic ratio remains the same. One such case is test cross. In this chapter we will see at another condition also. So here it is in the test cross, whether it is monohybrid test cross or it is dihybrid test cross, the phenotypic as well as genotypic ratio, that will remains the same. For a monohybrid cross, the test cross ratio will be 1:1. So three ratios are here in this monohybrid cross. One is phenotypic ratio, that is 3:1. Genotypic ratio is 1:2:1 and test cross ratio is 1:1. Memorize. Now comes the back cross, that is crossing of an F1 hybrid back to its dominant parent, that is called as back cross. Now we can see what is the result of a back cross. the F1 hybrid that is crossed back to its dominant parent. So, here in this particular case, the dominant parent is tall and F1 hybrid is also tall. So, tall into tall. This is the F1 hybrid and this one is the dominant tall parent. Now, coming the genotype, since it is F1 hybrid, be careful, it will be a heterozygous one. So, capital T small t and this one is capital T because it is dominant parent and pure line. So, it is homozygous. Gametes capital T small t like this. Suppose we are representing it in the Punnett square. The male gamete capital T, here also capital T. This is the female gamete capital T small t. Capital T one union, the other combination is capital T small t, here it is capital T, this one is again capital T and small t. Now, what will be the phenotype? Here it is homozygous tall, these are heterozygous tall. That means all the progenies are tall in phenotype. Tall in phenotype means it is dominant. So, again coming the concept, in a back cross, all the progenies produced will be dominant in phenotype. That is the common conclusion. But in test cross, we will get one dominant is to one recessive. That is 1:1 ratio of phenotype as well as genotype. Mendel's work remains successful. So, what were the reasons for his success? First of all, it was the selection of an ideal or a suitable experimental material. You all know that the experimental material is Pisum sativum, that is garden pea. And second, intelligent decision that he selected, that was selection of a true breeding plant for his hybridization experiment. You all know that true breeding? Yes, it is pure line homozygous plant. The another important concept is he studied only one particular character at a time. There were 7 pairs of contrasting characters and out of that 7 pairs at a particular time he completely studied the inheritance of a single character at a particular time. And moreover, he kept the accurate records of his hybridization experiments for further reference and study, and he used some of these statistical methods and law of probability to analyze his results of hybridization experiments. So, in this way his work, breeding experiment, the hybridization experiment was a very successful one. Now come the question, what is meant by a reciprocal cross? Mendel conducted original cross by taking tall plant as male and dwarf plant as female. but he also conducted the reciprocal cross. Now, what is the need of this reciprocal cross? What is reciprocal cross and what is the need of it? The reciprocal cross is a cross in which the sex of the parents are reversed from that of the original cross. Here you can see original cross, tall plant, that is considered as male, which is crossed with dwarf plant, that is taken as female. So, this is the original cross done by Mendel. Now comes the question, reciprocal cross. Here the sex of these parents are reversed. That means the tall plant, that we will consider as, yes, female, and dwarf plant we will consider as male. Only this thing. Here you can see the difference in the sex of these individuals. The sex of the crossed parents are reversed from that of the original cross. Now this one we will call it as reciprocal cross. Now what will happen if we are doing like this? If in the result will be the same. We can do, here it is female, here it is dwarf plant is taken as male. So here comes emasculation. Tall plant with flowers are emasculated for converting it into a female flower. So this is the phenotype. Now comes the genotype, capital T, small t, small t. Gametes, capital T, small t like this. Now we can use the cross pollination, hybridization, collected the pollen grains from this dwarf male plant, dusted on the stigma of the tall female plant. The result will be capital T, small t, same as that of the original cross. You can see here, same as that of the original cross, that you can see here. The F1 hybrid phenotype again tall. When we are again doing this selfing of this plant to produce the F2 generation, see selfing of F1, capital T small t into capital T small t, gametes capital T small t, capital T again small t. We can represent it in the form of a Punnett square. Like this you can see the result. No difference, same result we will get, 3 tall and 1 dwarf. That is again 3:1 ratio. Regarding the genotype also, we will get the same result, 1 capital T, 2 capital T small t and 1 small t small t. So again it is 1:2:1, no variation from that of the original cross. So Mendel also want to confirm whether the results remains the same or it varies. So for that purpose he used this reciprocal cross. So the results and the ratios that will remain the same. Now comes the laws framed by Mendel based on his hybridization experiments, breeding experiments, that we will name it as Mendelian laws. So, the first one, that is law of unit character. From the very beginning itself, it was told that each and every character is controlled by a pair of factors. For example, here also you can see the tall character, that is controlled by a pair of factor capital T capital T. Dwarf controlled by the alleles small t small t. So, each and every character that is controlled by a pair of factors or alleles. So, that particular law, that is known as law of unit characters. For example, if it is a purple flower color, it is controlled by capital P or it can be controlled by capital P small p. One is homozygous and the other is heterozygous. Now, coming to the second law, that is law of dominance. You can see the law of dominance here. Why this progeny F1 hybrid becomes tall? That will say the answer of law of dominance. Here you can see two dissimilar alleles present in this hybrid individual. Out of these two dissimilar alleles, one allele is dominant and it will express its character, it will produces its phenotype, and the other one get suppressed. Here capital T is the dominant allele that get expressed and small t gets suppressed, which is recessive allele. So, the allele that get expressed, it produces its phenotype, that is known as the dominant allele, and the allele that get suppressed, which will not produce the phenotype, that is called as recessive. So, this particular law, based on this particular law, the progeny, this F1 hybrid progeny becomes dominant, tall in phenotype. So, in the coming genetics problems, along with that problems, there will come the question, state what is the law based on this particular problem. So, there you can analyze, one is law of unity character, second thing is law of dominance. Now, comes to the next one, that is law of segregation. Here you can see these two alleles, they are seen together in this particular organism. Here also in the tall phenotype controlled by the alleles capital T, they are seen together. Here also small t alleles, they are seen in pairs. So, these alleles during the gamete formation, you can see here clearly allelic pairs will separate, they are segregating and moving to two different types of gametes. Here also the capital T and small t alleles, they are seen in pairs, but during gamete formation they are moving to different gametes. And you can see in a particular gamete, there is only one particular allele of a particular trait. Not both the alleles are going together in one particular gamete. Only a one gamete that contains only a single allele for a particular trait. So, the gametes are pure here, either it may be showing dominant allele or it may be showing recessive allele. So, this particular law, that is known as law of segregation or it is known as law of purity of gametes, because the gametes are pure here, either it may be dominant or it possesses a recessive allele. So, this is yet another law based on monohybrid cross, law of segregation, or at another name is law of purity of gametes. Why this law is known as law of purity of gametes? Because as I have already told, the gamete contains only one allele for a particular character. Now comes the next and the last of the Mendelian laws, that is law of independent assortment. We will see this particular law in detail and we will explain it in detail in the next session, that is in dihybrid cross, because this particular law is based on dihybrid cross, not based on monohybrid cross. In this present session we have dealt with monohybrid laws and monohybrid cross. Now it is the time for review questions. You have attended and listened the session very well. Now give me the answers. Here is the question for you. Mendel crossed a true breeding purple flowered pea plant with a white flowered one. What will be the nature of F1 progenies? When the F1 progenies are self pollinated, what will be the phenotype and genotype among the F2 progenies? This is the question for you to answer. So, here I will help you in answering the question. In this problem, it is already mentioned that the character is purple and the other one is white. So, from this hint, it is easy for you to identify what is the type of cross. It is a monohybrid cross, and here already it is given the clue that the parents are true breeding. True breeding means homozygous. So, we can solve this problem in the following way. Parents, male and female. Phenotype, purple, white. Genotype, since it is true breeding, it is homozygous. So, we will write it as capital P, capital P. We have used the letter P because the dominant character begins with a P. The small letter small p is meant for white. Now, gametes, capital P, and here it is small p, small p. F1 generation, it is capital P first followed by small p. The progeny is purple in phenotype. This is the F1 hybrid. So, here in the question it is already asked what will be the nature of F1 progenies. So, here comes the answer. Its genotype is capital P, small p, and the phenotype is purple. Now, here it is again asked in the question, what will be the nature of F2 progenies, phenotype as well as genotype. That means, we have to show the F2 generation also. So, F1 on selfing, we will represent it as capital P small p into capital P small p. One consider as male and the other is considering as female. Now, gametes, capital P1 gamete small p at another gamete, here also like this. Now we have to make the Punnett square for representing the F2 generation. Male gamete on the vertical side here, capital P, small p, and here is the female gamete, capital P, small p. So, among the progenies, the combination here is capital P, it is capital P, small p, here also capital P, small p, and this is small p, small p. See the phenotype, this one is purple, these two are also purple because capital P gene for purple is dominant over small p gene for white. So, total number of purple progenies will be 3 and white, that is only 1. So, here the phenotypic ratio, that is asked in the question, what will be the phenotype in the F2 progenies. So, your answer comes here 3 purple is to 1 white. Now comes the genotype. One is having capital P genotype, that is homozygous for purple, and 2 individuals are having capital P and small p genetic makeup. The other one is having small p gene constitution. So, here the genotypic ratio is 1 is to 2 is to 1. So, this is your answer for this particular problem. Now, here are some questions from your NCERT textbooks. Using a Punnett square, work out the distribution of phenotypic features in the first filial generation after a cross between a homozygous female and a heterozygous male for a single locus. Since it is mentioned single locus, heterozygous for single locus, that means F1 hybrid. And it is again mentioned that homozygous female. So, from these two conclusions, we came to the point it is a monohybrid cross. We can consider the cross tall into dwarf. A character we are taking, for example, the contrasting characters we are considering is tall and dwarf. And based on this particular character, we can show this cross with the help of a Punnett square. So, here one individual is heterozygous. So, the answer for this question number 2 is tall into dwarf, capital T, small t tall, because it is already mentioned it is heterozygous for a single locus. So, it is capital T small t. Here it is homozygous, that is dwarf. Dwarf is homozygous and recessive. Heterozygous individual must be taken as male. So, this one will be the male and this will be the female parent. Now, the gametes are capital T, small t, here it is small t, and we have to represent again it in the Punnett square. Female gamete small t, male gametes capital T, small t. So, capital T, small t, again capital T, small t, and these t are small t, this one also small t. Here comes the phenotype, 2 tall and 2 dwarf. So, again it is equal to 1:1 ratio. So, it is a test cross that is asked in the question. Your third question is, a diploid organism is heterozygous for 4 loci. How many types of gametes can be formed? Heterozygous for one locus, then it produces 2 gametes. It can be calculated by using this formula 2 raised to n. Here n denotes the number of loci or even number of contrasting characters. Since it is 4 loci, it is 2 raised to 4. That means 2 into 2 into 2 into 2, that equals 16 types of gametes are produced here. Now, this is the question for you to think about and produce the answer. A pea plant producing axial flower is crossed with another pea plant producing terminal flower. Among the progenies, 45 plants are producing axial flower, whereas 48 plants are producing terminal flower. What is the type of inheritance noticed here? Write the genotype of the parents and work out in a Punnett square. So, remaining we will see in the next session. Thank you.